Professor Biankin is a Cancer Research UK clinician scientist and Wellcome Trust senior investigator. He is also a fellow of both the Royal Society of Edinburgh and the Academy of Medical Sciences. At the helm of the Wolfson Wool Cancer Research Center, Professor Biankin leads a team dedicated to the pursuit of precision oncology. His leadership extends to national and international consortia focused on cancer genomics and therapeutic development. With over 160 articles published in major journals, including groundbreaking contributions to cancer, genomics, and precision medicine, Professor Biankin's impact on the field is undeniable. Currently, his efforts are concentrated on implementing precision oncology practices in health systems worldwide, demonstrating his commitment to translating research into tangible improvements in patient care. Excitement that we welcome Professor Andrew Biankin as the state-of-the-art lecturer for the 2024 HPBA annual meeting. Please join me in extending a warm welcome to Professor Biankin.
Thank you, Lynn, for a very kind introduction. Thank you, Peter and Sean, for the opportunity to share some of the things that we're doing and some of the things we'd like to do in the future. So just following on what Nancy was talking about, 50% of any financial cost of any health service is spent on the last 9 months of life. That to me tells me we're not doing it particularly well. We're developing new and new therapeutics, new and new procedures, which just cost more and more and more and more. But the problem is there's a lot of waste. Don't work. Who in this room has hypertension? Right. Oh, come on, there's more of you, at least 30%. Okay. Who's on one drug? Keep your hands up. Keep your hands up if you're on 2 drugs. Keep your hands up if you're on 3 drugs. Which one of those is actually working? It's not the combination. There you go. I see patients in the UK, and drugs are free there, so anybody can get drugs. You don't have to pay for them. There are people on 25 medications. How can they manage breakfast after that? It's impossible. There are 90-year-olds on statins. I'm thinking, for goodness sake, we've got a massive amount of waste. So how do we change that? And how do we do that? These are my disclosures. They used to be better, but I'm working on that. So pancreatic cancer first, and I'm going to talk about cancers in general, but pancreatic cancer first, we're stuck. We have a bunch of drugs. We have lots of drugs to choose from, but their effect is quite small. And we're sort of married to, we've got to give them FOLFIRINOX. We've got to give them FOLFIRINOX. And most of the time, it doesn't really work. And then of all the people that do get first-line therapy, only 50% of those. But we don't have a denominator. There are data out there that in well-developed countries, only 50% of patients with pancreatic cancer actually receive any treatment at all. And then with drugs in pancreatic cancer, we have to do it right at the end, where the last 12% that might get a 3rd line of therapy. So we're stuck. How do we change that? And this is not just the case for pancreatic, which is a great example of it, but we're doing it across all cancers and, in fact, chronic diseases. Biology. We'll understand all the biology, and it's going to be fantastic. We're going to find all these new targets. We're going to make drug towards them when we do that. But every drug I've looked at, this is a pure inhibitor of this kinase. And then you look at the kinome map, and there's another 30 lighting up like a Christmas tree. So we don't really know exactly what they're doing. The FDA says, oh, we need to see the biology. 90% of most of it's not reported very well. You can make up any sort of story of biology if you've got the right articles. So, I mean, is biology the answer? Yes, sure it is to a certain point. But do we have the time to do the 3,000 years of experiments that are required to understand biology of each gene, and then the interactions with scale tens and tens and tens of billions fold? The answer is probably not. But can we shortcut that in a particular way? Now, yes, we'll look at the transcriptome and variants of subtyped pancreatic cancer based on statistics. And there's an obvious reason why there's two differences, because the mutations in each one of them are different with the epigenetic changes. So we'd expect a massive change in transcriptome. There is some underlying biology, different subgroups. We still treat them the same. And if you give an inhibitor to one pathway, it might be beneficial to one and detrimental to another. So in a clinical trial with these targeted agents, you might be benefiting 20%, but making it worse for 10%. So you get a 10% benefit. We assume that it works. Maybe we're doing some harm. So in the UK, we thought we need to do something. We need to push this stuff along. So we set up a platform called Precision Panc. 30. Really, the principle being is to molecularly profile all our patients as they come in through the door for the trial, because we couldn't afford to do all of them. Identify particular targets through a large diagnostic panel, and then allocate them either onto treatments and trials. And this has been tough. Probably the best success is that before we started this, almost no patients got a biopsy in pancreatic. You might get a bit of a brushing, you might get a bit of an FNA, and sort of look at the scans and go, oh, yeah, it's pancreatic cancer. And maybe the results in something that you see, either you've done a really good job, you've successfully treated chronic pancreatitis with gemcitabine. So now each patient gets a biopsy, and we've trained all the sites across the UK to do an endoscopic ultrasound guided core biopsy, but it's a micro core. It's only 21 or 22 millimeters, and we can do a large gene panel and a transcriptome on that single small sample. So there's a master protocol, and we had to have an all-comers funnel. You need to offer every patient some opportunity. And then in the second line is the field where you start to do more targeted things. And that's been difficult because pharma, pancreatic cancer, the graveyard of therapeutic development. It's been a challenge. A number of studies we've pretty much got up and running, and the drug gets deprioritized. A biotech company builds up value in their assets by saying, hey, yeah, we're going to run this trial, and then they're acquired by another company, and there's gotcha every step of the way. And most of it isn't scientific. Most of it is really societal. That's been tough, and we're pushing that along. But the one I just mentioned today is the all-comers study, just to keep the pathway flowing with all the molecular data. And that's something that we'll read out in the next 12 to 18 months. Really comparing FOLFOXA versus Abraxane gemcitabine. Not a great innovation. You just take your irinotecan out because we know it doesn't work that well, and you can't have FOLFIRINOX-A because you'll completely blow up your patients. So that may give us some information that's useful, but more importantly, each one of these has got a genome and a transcriptome. So we'll get some insights there. So how do we really shift the needle? We're not shifting it for pancreas. Incremental gains, but I suggest that if your 2-year survival is less than 10%, you still don't really have a standard of care. You're sort of shuffling deck chairs on the Titanic to a certain point. But some of those drugs, Abraxane and gemcitabine, FOLFIRINOX, stabilize the disease, makes patients healthier, last longer, and then we can conduct more clinical trials on them. So can you believe that not that long ago, we used to bleed people, do other barbaric things to them to treat them? How crazy is that? I mean, we used to do that. And in fact, we still bleed people to treat them, but only in 2 diseases, hemochromatosis and polycythemia rubra vera. So bleeding does work, but not for everybody. And what's going to happen in 50 or 100 years' time? The patient would say, can you believe that not that long ago, we gave toxic chemicals to people to treat diseases without knowing if they would actually work for them or not? I mean, like, if you get FOLFIRINOX, you're poisoning 70% and maybe giving 30% some benefit. What we can't do is predict things for an individual person. And that's what the patient wants to know. That's what the doc who's treating the patients wants to know. These sorts of questions, we can answer them on average, but what use is that to you? There's no such thing as equipoise for you when you're a patient. What's it going to do for me? And how do we unlock that? We need data. So we now run the International Cancer Genome Consortium, very much about clinical data, and that's tough. We've got a number of countries on board, data coming in. Molecular data is easy now. You do a genome, upload it, it's fine. But getting the clinical data is the toughest component. As part of that, we've partnered with the Lancet Oncology to write one of their commissions about how we might advance this precision oncology, including low to middle income countries, and how we can unlock that and better treat our patients. So the bit I really want to talk about is opportunities. AI, we all talk about AI. Everybody's really excited. Now everybody thinks it doesn't work, but it does work. It's just probably not as good as it could be. And maybe it's positioned in the wrong spot. Drug design, most of the trials I've gone through haven't succeeded. Where do we go with that next? It's very useful in the financial sector, useful in other arenas. And by the way, AI is just trial and error really, really, really quickly. But you use different analytical ways to do that. They can be linear, neural network type, or Bayesian. And this is important. Hard to get patients onto the control arm of trials. Patients will shop around. So what about synthetic controls? I mean, for years we had gemcitabine as the comparator, the most stable thing in pancreatic cancer. Median survival was always, 6 months. And we had to keep running things on the control arm. Why? Is there that much variability once you've got that historically? Now there are provision of synthetic controls, and the regulators looking at them very closely, age, disease matching patients that are the control arm of that data. Probably not real world data, although there are the companies making a lot of money on that. You want to get a synthetic control out of one of these companies for 30 patients, it's upwards of 5 million to do that, because it's going to cost you 10 times more to run those on a study. And what about the patient being their own control? These N-of-1 type studies. In fact, that's how cortisol was discovered. So a guy called Hench in the 50s, and you can do this in chronic disease, he extracted, I don't know how many thousands of adrenal glands from the abattoirs and gave it to some of his rheumatoid arthritis patients. And these patients were completely debilitated. They couldn't get up out of their chair. So we did almost the Cox postulates of chronic disease. So he gave them injections of this extract. And after about a week, they got up, started walking around, really back to being almost normal again. Then he did the thing he had to do, which is stop the treatment. And within a week, they were back and they couldn't move. So we can't do that with cancer patients. And even if we do it, we don't believe it because, oh, the CA19-9 went down. Oh, what does that actually mean? Oh, this marker went down. Oh, ctDNA, maybe there's something in that, surrogate endpoints, perhaps. As I'd mentioned, trial and error oncology. This is what we do. This one has the highest chance of response. On average, you're going to get that one first. 3 months goes past, you do your scan. Ooh, is it progressing? Is it not? We'll give you another 3 months of that. Get another 3 months, and you look at the scan and go, oh, shit, it's not working. What do we do now? Panic, panic, panic. Oh, second line. Same thing again. Then the third line, we have to do this because this is how we're told to do it. You have to give this first line, you have to give this second line, you have to give this third line, but you can't predict who's going to respond ahead of time. And this gives high toxicity and side effects, high cost of care, and nobody really measures this in health economics. There's lots of health economic activity, but there's no data. Yeah. How can you get the data that says this patient had an excessive admission, spent 3 weeks in intensive care because he had this toxic reaction? What was the whole total cost of that? And for most, it's limited benefit, and patient outcomes are still overall remaining poor. So we moved to something, and this is a company that I collaborate with and subsequently have invested in as well, using astrophysics technology. So any astrophysicists in the room? Okay. Any surgeons take astrophysics as a hobby? But maybe a bit too geeky. Oh, there's an arm up the back there, I think. Okay. So gravitational lensing is how gravity acts. Fragments of activity, lots of different interactions going on, lots of different variable bits of data. You can predict the mass of dark matter by the way things are bent by light. You can see stars, the same star 5 times behind a galaxy cluster because the light's bending around it from both ways. So you can see the same star 5 times. To measure that and predict that and discover black holes takes a lot of complex mathematics, a lot of understanding of these different things. And it turns out it's very similar to biology. You look at these different interactions, how do these systems work? And so we adopted that, evolved that in order for it to work in biology. And at first I thought, oh yeah, guys, you're a bunch of geeks. What are you going to, really, is it really going to work? So we tried building digital twins. And the critical aspect of this, it's a twin of an individual patient. So you construct whatever omics data you can get, bring it together with the clinical data, and you can start to potentially predict survival and response, et cetera. And there's a lot to that, but I won't go through all of that. I'll just give you one component why I'm so excited about this. You can bring them together and start to do in silico clinical trials based on these predictions. You need to get reasonable data in first because the algorithm needs to learn. It's Bayesian, so your pre-test probability goes from everything to dun, 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 dun. You can start to predict things in smaller numbers, which is kind of cool. And you can start to compare these things in silico. So I thought, guys, yeah, that's great. Looks exciting. Prove to me that it works. So we gave them 8 clinical trials for them to predict. They're guys that sit in the computer room. They don't even know what a clinical trial is most of the time. So it's like, can you predict these trials? And all these 8 trials in different diseases in increasing complexity, without knowing what the results were, they were able to accurately predict what this trial would be like based on completely different data. Not the data from the original trial, because of course you never get that. That's buried somewhere and you can never find it. But this is completely different cohorts of patients. And I thought, well, maybe there's something to this. So predicted 8 out of 8 correct, positive or negative, et cetera. So can we use this to better select patients for existing treatments? Most of the data that is publicly available or accessible is about standard of care chemotherapy. And we waste a lot of that. So here we have an example. 4 possible therapies, maybe they're first line, second line, third line, fourth line, or in lung cancer, chemo in combination with a platinum, you've got a choice of 6. Which one do you use? Well, you go with the one that's got the greatest odds. But then let's look at the individual. What would you do in that context if you're Mrs. AS? You see these, would you say, I still want to go with the first treatment, or maybe I'll do the third one, and then it's different for the next person, different for the next one, and different there. And that's me at the end there. I've got almost nothing for my pancreatic cancer. And I think, well, Paya, how much would you have spent on me? It's probably $1 million to give you all these treatments. Let's do a deal. Give me $500,000. You keep $500,000. And I'm going to go buy all the DRC Le Montrachet I can get and have a damn good time. Doesn't represent any organization. Develop it. What's a new model? I mean, if you were doing this, I can't go back a slide. You can imagine a day when you're sitting with a screen in front of you, data is imported from the various labs, you enter the clinical data, and out comes a probability of response for the patient sitting in front of you. Of course, there's a way to go and validation and testing, and we're talking to consortia about doing that. But how does this work in drug development? And this is where AI probably doesn't work that well. The current model is, we know we need to select patients for treatment better. Everybody knows that. Pharma knows that. Biotech knows that. We need a biomarker, need a biomarker, need a biomarker. We know the probability of success increases dramatically if you have a biomarker of therapeutic response. But how do you get there? So you do all this deep omic data on your preclinical, your early clinical studies, and then you try and pare it down into a couple of genes or maybe a signature that says, okay, we can do that. And we have examples of that where they have worked to a certain degree, Oncotype DX and maybe a few others, and Caris has one for colorectal cancer, but they're not great. They make a bit of a difference. And then you resolve this, and then you run your clinical study, and it might be positive, but most of the time it isn't. And even if it is positive for all comers, the incremental gain of your biomarker is so small that nobody ever uses it. But what if we start to use this further down at the bedside? Do generic type of tests, and it's a whole exome and a whole transcriptome, and it's available. Companies like Personalis, Caris, $5,500 for both from a standard routine biopsy, cheaper than Foundation Medicine list price. You're already spending that money anyway. So I asked the guys the question, so well, why aren't these linear biomarkers working? So digital twin is the Bayesian algorithm. And these are the examples of 4 of the best linear algorithms to predict. And you can see none of the 4 predict. And no matter how big these signatures are, and you can see the graph on the right, the predictive value, even if you include all the genes using linear approaches, and this is non-interactive Bayesian modeling, you can see you get 0.55 at best, 0.5, which is a guess. So we can never get there. We might get lucky in a few cases, but as a general rule, we're not going to get there. So that model is just not really going to work. But what can we do if we do it slightly different? Let's instill AI not there and then lose the power of AI, because if you're going to make something into a simple biomarker, you don't need AI to do that. You can just look at it and go, oh, I've seen cyclin E amplification 15 times out of the 20. That looks like a good one. Don't even need statistics. So can we do that? And how can you use that in clinical development? Can you generate synthetic controls? This is one example. So you've done a study, right? Sorry, I meant to say the ability to predict your treatment response to standard of care can completely change the way you develop drugs. So you've done this study, single arm, still do these studies, 30 patients, expansion. I don't want to go into the last phase because these patients, have had multiple pre-treatments. It's just really difficult. It's a tall bar to get past. Great for immunotherapy because you've really messed the tumor up, so you're increasing that. But for small molecules, it's more difficult. And you look at it, okay, this curve, 20.7 median survival. And this is standard of care plus the new treatment. And you look at your historical study and you think, well, all our controls have been, in all our previous studies, there's been median survival of 20. Bennett, it's a no-go, clear no-go on this. We're not going to take that forward. But what if you looked at these individually and you predicted what the response to standard of care would be? So those little lines with the dots on them are the predicted response with a 95% confidence interval. Single arm study, standard of care plus novel therapeutic. The little bars are standard of care prediction. And the red star is the actual survival. And you think, well, some of those are benefiting pretty well. They're outside that sort of level. Maybe there's something in this. All right, let's just compare them together. To compare the prediction to your trial. And you go, it's not looking that great, but there's a bit of a bump there. So what if we just take those 10 patients and look that they've got responded, they responded, bang. Enormous difference, sure, small numbers, but that information from there, based on an exome and a transcriptome, or even a large panel and a transcriptome, which are available and clinical grade, clear approved, you've suddenly got a third of patients with this dramatic response. You take that into a subsequent study, take the information from that, and you've got a successful drug that's going to be effective through a much higher bar and much more beneficial. So every cancer patient should have a broad test, not just the focused one, because then you instill your AI at the bedside or at the recruitment side. The algorithms, they're in front of you on your computer, you upload the stuff into some clinical data, press a button, say, right, okay, this is the right thing for this patient, either for current standard of care or for a new drug. So the companion diagnostic is not the assay. The assay is generic, it sure includes the assay, but the analytic is the companion diagnostic. And envisage that in the future there'll be multiple different companies making different analytics to try and push this thing forward. So the new model would be, let's use AI and let's not lose its value by ignoring all of the detail. Let's understand what the AI does. Let's use it better and go from linear space to interactive space, which increases your dimensionality, allows you to have much, much better false discovery rates, takes away all that Bonferroni adjustment challenges that you get, the multiple hypothesis testing challenges you get in linear stats. And it's there. It's cheaper than a Foundation Medicine test. So with that, I'll end. A whole number of people to thank. I've spent a shed load of money in my life, and this is my my supporters. And thank you for listening.
Professor Bianchi, don't go anywhere yet. On behalf of the AHPBA, we would like to thank you for delivering that insightful discussion on an innovative and really important practical solutions to bringing personalized treatments for patients with cancer. Thank you very much.
My pleasure. Thank you, Nick.